This is Laura Fulford welcoming you to this Bite-Sized Bio web seminar, which today is sponsored by GE Healthcare. Today's presentation is titled Practical Super Resolution Imaging, SIM Capability, Sample Prep and Analysis. This webinar is being presented by Patrina Pellett. Patrina received her PhD in the laboratory of James Rothman at Yale University. She specialized in the development of live cell super resolution imaging methods to study intracellular trafficking. After her graduate work, Patrina joined GE Healthcare as an application scientist for GE Super Resolution Microscope, the OMX. Her focus is providing researchers and OMX users with training, technical expertise and support for super resolution, TIRF and conventional imaging experiments. We will have a brief question and answer session after the presentation, so please type any questions that you have into the questions box, which appears on the right hand side of your screen, and I'll put them to Patrina at the end. So now over to you, Patrina, for the presentation.
Oh, sorry, is my screen showing? Can everybody see that? Or
Yep,
Laura, is it? Oh,
yes,
okay.
that's absolutely fine.
All right, thank you. Sorry about that. I wasn't sure if my screen was actually showing. Welcome. Thank you everyone for calling in or joining the webinar. Today I'm going to talk about one specific kind of super resolution imaging called structured illumination microscopy. My goal is to really get you to start thinking about how imaging with SIM could potentially further your own research project. So I'm going to go over a very brief abbreviated version of theory. We'll talk about some sample prep tips and also some analysis tips and how to avoid the common SIM artifacts or some of the common artifacts. I am an application scientist for the OMX, so I spend a lot of time thinking about the OMX, but I tried to make this seminar a little bit more general so that other non-OMX users can get something out of it as well, if there are any on the line. Okay, so the plan for today is to go over a brief introduction of light microscopy and what are some of the advantages and disadvantages. And then I will give you a very, very short version of SIM theory. This is really going to be a version that you could go and tell your lab mates about when they ask you how this technique works. Then I'm going to show some of my favorite SIM examples and where this really allowed researchers to see something that they couldn't previously see with conventional imaging techniques. Then we'll go over some of the sample prep recommendations and then finish up with some of the common SIM artifacts, and then I'll make suggestions on how you can fix those. So if you're not really don't really know a lot about SIM or are using it a lot, that's probably a really good time to drop out of the webinar. Okay, so here's a preview of some of the stuff to come. This is just giving you a little bit of teaser of some of the great images that we can get from structured illumination microscopy. This is a 20 micron thick stack of a yeast biofilm, and this group is interested in understanding how the single cell forms of the yeast form this infectious mycelium. And notice as this screen is rotating or the video is rotating, you can start to see some of these little bud scars. They couldn't previously see this with their the current conventional imaging techniques they were using. So what are some of the advantages and disadvantages of light microscopy, and why is it such a widespread technique, especially in the biological sciences? Well, the main reason is because it actually allows us to see what our sample looks like. And this makes it a lot easier to understand the function of the particular protein or structure that we're studying. So with light microscopy, we can visualize our protein in both 2 and 3 dimensions, and we can start to get an understanding of how our protein of interest relates to other proteins found in the cell, and do co-localization type analyses. We can additionally determine the shape or morphology of the structure that we're interested in. And one of the biggest advantages of light microscopy over other conventional visualization techniques, such as electron microscopy, is that we can actually visualize our protein of interest in living cells in its native environment and potentially in real time. So here are a few nice examples of 3D SIM images that were acquired on the OMX. These are winners of the GE image competition. So here we're looking at a mitotic cell with the microtubules and kinetochore stained. This is another image of a dividing cell, this time with the the DNA labeled in blue in the middle. The final image I'll show you in this beginning section is this image of microvilli from the Tiska lab at Vanderbilt. I really like this image. It looks like, it doesn't even look like a fluorescent micrograph. I think it looks more like art here. Okay, so what are some of the disadvantages of this technique? Well, the biggest disadvantage of light microscopy is that it's diffraction limited, or this is preventing us from viewing things that are much smaller than 200 nanometers. And I'll go into more details on this in a second. We also start to run into roadblocks from the actual microscopes that we're using. So sometimes speed and sensitivity of the sample or of the system becomes a problem, as does some of the limitations imposed by the sample. So I'm going to now transition to talking about the diffraction limit of light and the effects that this has on biological imaging.
So to illustrate this, I have a few cartoons that I'm going to go through. So when we look at a single fluorescent point source through a conventional microscope, because of the wave properties of light and how it interacts with the optics, this single fluorescent point source will always appear larger than it is. And in this particular example, I'm showing that it will come out looking like it's approximately 260 by 260 by 540 nanometers. So what does this mean in terms of biological imaging? To illustrate this, I've placed these red dots next to this diffraction limited volume. And this red dot is representing the approximate size of a typical secondary antibody. So what we can see from this diagram is that we can probably fit thousands of these secondary antibodies into this diffraction limited volume. What this results in is limiting our confidence in the actual precise spatial location of the protein or molecule that we're studying. The last example I'll show to drive this point home is with this following cartoon. So in this example, I have taken this diffraction limited volume and I've made this yellow to represent the co-localization that we would see through a conventional microscope. So in this example, we have 4 green markers, 1 red marker that would appear like it's co-localized with conventional microscopy. So because of the diffraction limit, we don't actually know its precise spatial location. So it might have this orientation, or these molecules might have this orientation, or they could have an orientation of something like this. What this means is that biological structures or proteins on biological structures can appear to be over, or that are actually over 100 nanometers apart, will appear co-localized. And this means that they can be on completely different structures. So this has led to a recent explosion in these different super resolution techniques that help beat this diffraction limit and help us visualize things that are smaller than 200 nanometers. So now I'm going to go over structural illumination, or SIM theory. And again, this is a very, very abbreviated version. And if I'm not clear about anything, or if you want more details, please ask questions at the end of the webinar, or feel free to reach out to me later on, or any of the local microscopy experts you have in your university or department. So to summarize, what we're doing with structured illumination is we are illuminating the sample with a striped pattern. We then shift this pattern through different orientations to create something called moire fringes. These moire fringes encode high resolution information that we couldn't previously detect with our microscope. We then put this into Fourier space and perform a bunch of math on this to extract high resolution information and get our super resolution information out. So this resulting image will have a 2-fold increase in resolution, both laterally and axially if it's a 3D image, and then just laterally if it's a 2D image. We'll also see a significant increase in contrast. And the biggest advantage of structured illumination microscopy is that it's compatible with standard dyes and fluorophores. So this technique is not relying on a photophysical property of a dye. So this means you can go ahead and try using all the GFP or RFP constructs or normal fluorophores that you have in your lab with this technique. Okay. So to demonstrate what these moire fringes are, I'm going to show you this image of these zebras. Now watch what happens when I take this striped pattern and rotate it over the zebras. You can start to see the formation of secondary patterns. So in this image, it's most apparent on the legs and then also somewhat on the backs. So now look what happens when I rotate this pattern a different way. We start to see different secondary patterns form. So here in the backs or in the head of the zebra in this orientation.
So how do we create this pattern? How are we actually illuminating the sample with this striped or sinusoidal pattern? So in the case of 2D SIM, we split the laser beam through a diffraction grating. We block the 0 order beam and we take the +1 and the -1 beam and focus this on the edge of the back aperture of our objective. And that's what I'm representing with this cartoon here. So these little dots are representing the back or the laser beam, and this gray circle is the back aperture. What this does is results in a striped pattern that we use to illuminate the sample with. So here, this is what our normal wide field image would look, and this is a puddle of beads. And then if we image that same puddle of beads with our striped structured illumination pattern, we get an image out that has a bunch of stripes in it. We can't just image the sample one time with this stripe pattern. If you recall from the zebra example, we had to shift that pattern to create those moire fringes. So with structured illumination microscopy, we do this by shifting the pattern through different angles and different phases. So that's what I'm illustrating with this particular figure. So here for 2D structured illumination, we need to image at 3 different angles and then 3 different phases each for a total of 9 images per optical slice. Importantly, when we're doing 2D structured illumination, it's important to realize that we don't get modulation, or we don't get the stripe pattern in the Z. So this means that we don't get an increase in resolution in the Z. Okay, so in comparison, for 3D structured illumination, instead of blocking that 0 order beam or the middle beam, we focus this into the center of the back aperture. And what this does is create a 3D structured illumination pattern that we use to illuminate our sample with. And this looks something like this. So this is an orthogonal view, like we're looking straight onto the objective. So our sample will sit here, and our objective would sit down here. So what's important with a 3D SIM versus 2D SIM is that the 3D SIM pattern has modulation or change in contrast along the Z dimension, and this allows us to get an increased resolution in Z as well. So with this method, it requires more images. For 3D structural illumination, we have to image the sample with our striped pattern at 3 different angles and then 5 different phases. So it's a total of 15 images per optical slice. So this is an example of a typical structured illumination experiment. So notice as we go through this Z-stack that the orientation of the striped pattern changes. So as this flips over, we can see that the striped pattern is in a certain orientation. Now it's shifted by 60 degrees, and the twinkling or sparkling effect that you see is the shifting of the pattern through those 5 different phases. So this is a figure to illustrate the differences in resolution that we achieve with 2D, 3D SIM compared to wide field. So in this leftmost panel, we are getting this is an image of a single bead, a green fluorescent bead, and we get about 250 nanometer resolution lateral, and then about 650 in the axial dimension. When we process this through a 2D SIM algorithm, we get about 115, 120 nanometer resolution, but almost no increase in resolution in the Z. In contrast, with the 3D SIM, we now get a 2-fold increase in resolution in the axial dimension. So this is something to keep in mind when you are considering doing SIM for your own research and trying to decide which form of SIM would be best for you. So to summarize, we illuminate the pattern, or I'm sorry, we illuminate the sample with a striped pattern. And if you're doing 2D or 3D SIM, this dictates the total number of images that you need to take per optical section. What this does is result in the creation of moire fringes, and if you remember from the zebras, these are these secondary patterns that form that we couldn't previously detect with our microscope. Another way to think about this is that we are downsampling frequencies that were outside of the bandpass region of our microscope into a region that we can now detect. We take this information and extract it out in Fourier space, and we get a resulting super resolution image out. So this is a table illustrating some of the advantages of SIM and some things that you may want to consider when you're deciding on which technique or which microscopy technique will be best to answer your particular question. So with structural illumination imaging, we get a 2-fold increase in resolution. This is wavelength dependent, so the more blue shifted wavelengths get better resolution. I'll oftentimes hear a criticism of SIM that it's only a 2-fold increase in resolution. And it turns out that there are a lot of biological sweet spots where this is giving just a perfect amount of resolution to be able to get more information, or the information needed compared to confocal or wide field imaging. As I mentioned before, this is compatible with all your standard probes and fluorophores, so you don't have to do a bunch of methods development or develop new constructs and whatnot to image with this technique. A huge advantage of SIM over the other super resolution methods is that you can do up to 4 colors in super resolution. Additionally, you can, depending on your sample prep, very large volumes are possible. So compared to the other super resolution methods, with SIM you can get 3D information or increased information due to the increase in Z resolution over several microns in the Z dimension. So I showed you that image of the yeast mycelium in the beginning of this talk. You can image tissue sections, also isolated structures like centrosomes. I'll show you an example of that. And then of course, culture cells. And one of the big advantages of this technique compared to others is that it is live cell compatible. So if you are looking at imaging by SIM microscopy and you know that your biological question will require live cell imaging, this is probably a really good technique to check out. So now I want to show you some of my favorite applications. So we'll begin with this image of a microglial cell in a thick brain section. So this is a 50 micron thick brain section that has been placed on the cover slip, and the microglial cells are imaged in red, and then there's a receptor that this group is interested in studying in green. So here, what we can see with the structured illumination image compared to the wide field image is that we get a significant increase in contrast, and we can start to see that the little puncta from the receptors now localize or appear to localize on the microglial cell. So here's a volume view of this, and in this view you can start to tell that there is indeed these green little puncta on the microglial cell, which you can't really tell by wide field microscopy. Here's an example of looking at centrosomes. So by wide field, we cannot make out the center or the ring structure of the centrosome, but with 3D SIM, we can clearly see that this does indeed form a ring structure. Here's an example of isolated centrosomes with 2-color labeling, and this is just highlighting that we can get super resolution imaging in multiple colors. So when we compare the wide field to the structural illumination image, you can see that we can make out this circular structure, which was not possible by conventional imaging. This is what I call a Z-stack movie. So here this is going from the top of the stack to the bottom, and this is in a cell expressing an outer mitochondria membrane protein and an inner mitochondria membrane protein. By wide field imaging, we cannot really make out the inner from the outer, but this becomes very apparent or easy to resolve by structured illumination. Here's an example of primary cilia. So by wide field, we cannot resolve this circular base structure, but by structured illumination imaging, we can clearly see that this is forming some kind of round structure here. Here's a volume view of this. So now I'll move on to some really great examples of live cell imaging with different forms of SIM microscopy. So here we're looking at a cell that is expressing life act. This is a 2D SIM image. This means this has one optical slice, and something that's very clear from this image is that you get a significant increase in contrast when you compare this to wide field. Here is some turf SIM imaging. So we're just looking at this focal plane close to the closest to the cover slip, and so notice that we can start to see structures that we couldn't previously see with the wide field, or I'm sorry, the turf image of this. Here are a couple more examples. So here we're looking at caveolin with the turf image over on the left-hand side, and then the turf SIM image on the right. And what we can see is that we can start to make out these individual little puncta, and I'll show you a zoom of this in a second. So here's a zoom of that box in before. Here we can see that these are probably individual puncta, but it's kind of hard to tell, but that becomes much more obvious in the structured illumination image. Here's another really great example of clathrin-coated pits. So if you notice in the turf SIM image, we can start to make out the ring structure of the clathrin-coated pit, but when we look at that by turf imaging, they just look like small puncta. And this was a time lapse where we acquired images every 30 seconds over the course of 3 hours. So we weren't seeing a lot of phototoxicity in this case. Now here are some 3D sim examples. So this is looking at a an ER protein called Sec61 beta. This is a 1 micron stack, and we imaged this for approximately 100 frames. So noticed in some of these denser regions, such as here or up at the top of the cell, that we can start to make out the fine details of the ER, where it's not really possible with the wide field image. So here's another image of Sec61 beta, just showing that, like we saw in the previous image, we can start to make out some of the fine ER structures that we couldn't really make out in the wide field image. And if we measure the resolution or the full width half max of some of these skinny parts on the ER, these are anywhere from about 120 to 150 nanometers. And this is right at close to the resolution limit for structural illumination microscopy. Also highlight that fast imaging times are possible. So in this particular case, for this 3D stack, we acquired a sim image in 1.2 seconds. Here's a very nice example of a long time lapse of two-color, or cells expressing two colors. So here we have mitochondria labeled or tagged with GFP in green, and then the ER in red. So notice how we can see these the ER dynamics as well as the mitochondria snaking around. And again, this is two-color imaging for 200 time points. The raw frame rate on the system for this particular example was about 100 frames per second, or imaging at 2.7 sim frames per second. So depending on your process that you're trying to catch, we might, with this technique, even though a lot of images are required, we might be able to get the temporal resolution that you need to address your biological question. The last example I'll show you is of a cell undergoing mitosis. So this is a thick stack. This is 7.5 microns. We're looking at the wide field image here on the left and the structural illumination image on the right. So this is projected into a single plane. And what we can see is that this cell is indeed undergoing mitosis, and this is often a measure of phototoxicity. So if you are imaging your cell and it will not progress through mitosis, this is usually an indication that there's too much light and some form of phototoxicity. And I'll highlight that this was almost 1,000 images per time point, and we were still able, or this cell was still able to go through mitosis. Okay, so now I'm going to switch gears and start moving on to more of the practical aspects of using this technique. I really want to emphasize that these are sample prep recommendations, so they're not requirements. I get a lot of people that say, oh, I can't image by sim because I did this or that. I think that if you have access to a sim scope and you think it might be useful for your microscope, that you should definitely just give it a try, even if your sample prep doesn't allow you to follow one of these recommendations. So a lot of these recommendations that I'll go over are for imaging on any high-end microscope. So you should be following these for your confocal microscope or any microscope that has a high NA objective. It's an industry standard that manufacturers of objectives are assuming that the end user is using a cover slip of a certain thickness. And this happens to be a number 1.5 cover slip, and this means that the thickness of the glass is approximately 170 microns thick. So again, the manufacturers assume that you're using this glass and make corrections in the objective according to that. So it's very important that you use number 1.5 cover slips. You can get different varieties of these. You can get standard cover slips or high precision cover slips. High precision cover slips have a smaller range of thickness and oftentimes result in less optimization, or the need for less optimization. So the following examples are to help you keep your sample within the working range of the objective. So any high NA objective has a very small working range. So we always recommend that you mount your sample on the number 1.5 cover slip directly. So this cartoon is an example of what not to do. So don't, if possible, don't put your sample on the glass slide. This introduces an uncontrollable variable into your experiment, and you can't really know what this distance is, and sometimes you can push your sample outside of the working range of the objective. This is not a typical problem for people that are studying culture cells. We find that this is more of an issue with tissue culture, and I'll go into that in a second. This is a cartoon showing how we recommend that you mount your sample. So most cell culture people do this already because you grow your cells on the cover slip and then label them that way. So please always mount your sample this way, and if it's possible for those of you that are preparing tissue sections, please always mount the tissue section on the number 1.5 cover slip. So depending on the thickness of your tissue section, this uncontrollable variable can really be a big problem for keeping your sample within the working range of the objective. You can also, something that we do on the OMX to help minimize spherical aberration and help get better structural illumination images out is we optimize the refractive index of the immersion oil we're using. So when you've mounted in this orientation, you can optimize like crazy for, let's say, a structure right here. But because of this uncontrollable distance, when you move on to image another spot on your sample, it might be at a completely different distance and you might need to optimize more. So if possible, please mount your sample in this orientation. And again, this helps keep it within the working range of the objective and making sure that we can actually image your sample. And keeping your sample closer to the cover slip is always better if you can do that, especially for structural illumination imaging. SIM is a wide field technique, so this means that it is very susceptible to background or out of focus light, and therefore we recommend that you do not use mounting media with DAPI in it. So this is just illustrating that you can potentially get a lot of background if you have DAPI in your mounting media. So most people think that DAPI only fluoresces when it's bound to DNA, but that's not the case, and it actually has a very broad emission spectrum, so you can get a lot of background across the spectrum. You can image DAPI by structured illumination, but we recommend that you do a separate labeling step and then wash it out. This is a figure showing the effect that hardening versus non-hardening mounting media can have on the 3D morphology of your sample. So these are nuclei. We're looking at the orthogonal view of nuclei imaged by structured illumination. And so what we can see is in mounting media that are hardening, such as prolonged gold or Movial, this can reduce or crush down the 3D morphology of your cell. This is not necessarily a deal breaker. It doesn't mean you're going to get a bad SIM image, but one of the advantages of SIM is that you get that twofold increase in resolution in the axial dimension. So we tend to recommend that if you can preserve it, that might be better. But again, if your biological question doesn't require 3D information, it's not going to be that big of a deal. So to summarize, in general, SIM has the least strict sample prep requirements of all the super resolution methods, and I often recommend to people, if they have access to a SIM scope, a good place to start is to just put on your sample that you've already prepared for the confocal, and then see how that turns out, and then go from there to see if you need to make any modifications. Always try to get your sample as close to a number 1.5 cover slip as possible. Another issue that we can run into with SIM imaging is that it requires a lot of images, so photo bleaching can be a problem. So try to use the most photostable dyes possible, and also consider using mounting media that contain anti-fade agents. The last recommendation I'll make, and I'll talk about this more in a minute when we get to the artifacts section, is that you should try to minimize the refractive index mismatch in your sample, and this can really help with the quality of the SIM reconstruction. All right, so now I'm going to transition, and I'm going to briefly talk about 3 of the main artifacts that you'll see in structured illumination images and how to fix them. So the number 1 artifact that I run into with this technique is an artifact called haloing or doubling, and this is caused by spherical aberration, and this can be induced by refractive index mismatch. So what I'm showing you here is the same cell imaged with 2 different immersion oils. So in the case where this higher oil with a refractive index of 1.528, we can start to see all these kind of reflections or weird things coming off of the side of the cell. In addition, the structure in the center looks kind of blurry. Now notice when we use this lower RI oil, we don't have this effect. So this kind of haloing or doubling effect is caused by, or can be caused by, refractive index mismatch. So what we recommend doing on the OMX, and you should be doing this for your other types of imaging as well to help minimize this effect, is to match the oil or use a immersion oil with a refractive index that minimizes this effect. So what do I mean by that? So here I'm illustrating a point spread function of a bead imaged oil with a refractive index of 1.526, and what we can see here in this point spread function is that we have a lot of asymmetry. So we have a bunch of this flaring up towards the top. Now, when I am talking about a matched point spread function, I'm referring to this scenario on the right, and you'll hear a lot of people talking about this. So the characteristic that we're looking for is the same amount of flaring on the top as on the bottom. So what happens with the SIM algorithm, especially in 3D, what we're doing is we're taking optical sections, so we're imaging, let's say along here, we acquire image here, acquire image here, acquire image here. The algorithm assumes that you have this orientation, or that you have a perfectly matched point spread function. So when it detects this light up here, it thinks that's real and it reassigns it. So that's oftentimes why you'll see these reflections that look similar to the structure that you are imaging. Okay, so now I'm going to spend a few minutes talking about how to correct for this. So you assess if your immersion oil is the best for your sample by looking at the point spread function. So here's an orthogonal view of a point spread function with the oil severely mismatch. And again, mismatch means having asymmetry in the flaring of the point spread function. So in an ideal scenario, we'll have the same amount of flaring on the top and the bottom. The general rule of thumb is that if your flaring is up or towards the cover slip, your oil is too high in refractive index and you need to go down. This is just a movie showing the lateral view of this, and what we'll see in this movie is that on one side we have these big rings or airy discs, but on the other side we don't. Okay, so let's say you're imaging, you're trying to match your refractive index. So you say, oh, okay, number 1.5 is too high, or 1.526 is too high, I'm going to try to go down. So here's a scenario where we're looking at the same bead but imaged with refractive index oil of 1.508. So this is now way too low. You see that we switched this orientation. So the convention here is that if the flaring is down or in towards the sample, the oil is too low and we need to go up. So here's the same movie, and what we're going to see is now that those big rings appear on the other side or in towards the sample. So here's an image of this matched point spread function. This is using a refractive index oil of 1.516, and here we can see that the flaring is the same on the top and the bottom, and we would consider this situation to be ideal. This is just the movie showing what this looks like if you were scrolling through the Z-stack. So we'll see the same amount of flaring on the top as we did on the bottom. So now what I've done is I've taken those images of those taken the beads in those different refractive index oils and imaged them by structured illumination. So, and then max projected that. So here is our oil that's too high, our oil that's too low, our oil that's just right. And notice how we've minimized that spherical aberration artifact or that haloing or doubling. Now, for those of you that are thinking, what difference could .002 make? I have imaged that same bead with a refractive index of 1.514 compared to the one that we thought was matched, and you can see that there is indeed a little bit more artifacts. So one caveat of this approach is that you can't simultaneously match the refractive index for multiple wavelengths at a time. So this means that some samples will have more some wavelengths will have more artifacts. Others. So in general, you can do one of two things to help correct for this. You can try to match your oil for the most red shifted, because generally speaking, the more blue shifted wavelengths are more forgiving. Or you can just match for the color you're most interested in, knowing that you may have some of that haloing or doubling artifact in the other wavelengths. So for those of you that are new to this, this is a table to help you kind of get a good start on which oils will be good for you to try when you're imaging your sample. So for imaging at the cover slip, depending on your wavelength, this is our general recommendation at 25 degrees. For every 5 microns that you go into the sample, you need to increase this by .002. And then the other general rule of thumb is to increase this .008 if you're imaging at 37 degrees. And If you still don't like this table or you don't want to refer to it, I'll point you out to the GE oil immersion calculator. So this is an iPhone app and also an Android app, and it's free on the App Store. And this allows you to put in different parameters about your sample, and it'll give you an estimate of which oil will potentially match your sample. All right, the second most common artifact that I see is caused by photobleaching. So essentially anything that gives an asymmetrical point spread function will give an artifact similar to the one that we see with spherical aberration. There's a general rule of thumb that you shouldn't have more than 30% of photobleaching across the stack. And then these are just suggestions down here in blue to help you, things that you could potentially try to minimize photobleaching during your sim experiment. So here's an example of what this artifact looks like. This is extremely dramatic. So there was 90% bleaching across the stack, but if you look at your sim reconstruction and you see these kind of weird square edges forming, this is indicating that you photobleached your sample and you need to try to optimize optimize the imaging conditions to minimize the photobleaching. So this is a screenshot of the OMX acquisition software, and the point of this slide is that I recommend you take a look at what your max intensity counts are when you start your image and pay attention to that over the course of the experiment. If you have too much photobleaching, try adjusting your parameters to help prevent that. So the last thing I'll talk about is the honeycomb artifact. This is probably the most feared of all sim artifacts. This is a regular or hexagonal pattern that forms. It's especially common on diffuse labeling or samples with bad signal to noise. This is the way that noise looks in sim images. So in wide field or confocal images, noise looks blurry. If we take that and then process it through the sim algorithm, this looks more structured. So this is an artifact of going from Fourier space into real space. So there are a couple things you can do to fix this, but one thing to keep in mind is that sometimes it's not possible. Not all samples are appropriate for sim imaging. Usually these are diffuse structures. So if you're trying to image cytosolic GFP, this sim is probably not the best choice for you. So the two things that you can do is try to increase your signal to noise without photobleaching. And there's a filtering or smoothing constant that's in the algorithm that can help with this problem. So I'm going to quickly go over those two. Here's an example of imaging with better signal to noise. So we have a signal to noise of approximately 11 here versus 57. We start to see this hexagonal honeycomby pattern in the low signal to noise, which is minimized in the higher signal to noise. So this Wiener filter constant is a smoothing function within the sim algorithm, and the user can decide what value to use to help minimize this effect. So here we're looking at DAPI nuclei or microtubules, the exact same data set but processed with a different Wiener filter constant. So with this low constant, we see more of that honeycomb effect, but as we increase that, that minimizes. Very important to keep in mind, when you are playing around with this and increasing this value, you are losing resolution. You are filtering out high frequencies. So to illustrate that, this is looking at a cross-section of one of these microtubules. I've drawn a line profile and fit this with a Gaussian to get the full width half max. And what we can see is that with the filtering, the higher Wiener filter constant or more filtering, you lose resolution. So in this particular example, we've lost approximately 27 nanometers of resolution. So please be very cautious of what you're doing when you are increasing this Wiener filter constant. So to summarize, I showed you that as we increase that Wiener filter constant, or the signal to noise ratio, the honeycomb pattern started to diminish. And the DAPI signal in particular looks smoother. But please, please keep in mind that you are removing information when you do this. So on the OMX, we recommend that you use values of .001 to .003. And for samples with bad signal to noise or diffuse labeling, we recommend using something, a Wiener filter constant of .005 or higher. So to summarize, sometimes SIM is not the most appropriate technique for your application, but I would first recommend trying to increase the signal to noise without photobleaching and then to play around with this Wiener filter constant. So to summarize, get your cover slip as close as possible, get your sample close to the cover slip as possible. Try to minimize spherical aberration by either matching your oil or in other options, and always try to get the highest signal to noise ratio without photo bleaching. So this is my contact information, my email, and my phone number. If you have any questions, please feel free to reach out to me anytime. If you want to learn more about our product, here's a link to our website. And I'm happy to take any questions that you may have.
Thanks, Patrina. That was a really excellent presentation. So we have a few questions from the audience now. If anyone else has a question, please feel free to post it in the questions box that appears on the right-hand side of your screen. We'll get through as many questions as we can today. If there's any questions that are left unanswered, Patrina will answer them after the webinar, and we will upload them in a PDF document to the website. Okay, so the first question. We can use the same sample preparation methods, e.g. buffers and fluorophores, for 3D SIM as a use for wide field fluorescence and confocal. Is this also true for localization microscopy?
That's not the case. So the sample prep or the dyes required for localization are different. So this is localization is relying on the dye's ability to blink. So there are some dyes that are better than other dyes. So for localization imaging, currently one of the best dyes is Alexa 647. And to get a good idea of what other dyes might you may be able to use for this technique, I would recommend looking at a paper. It's Dempsey et al. Nature Methods 2011. And this is from the Zhuang lab at Harvard. This has a great table illustrating the different properties.
That's wonderful, thank you. The next question is, why is SIM suitable for live cell imaging, whereas Palm and Storm are not?
That's a great question. So it really has to do with the temporal resolution. So for localization imaging, you need to collect thousands and thousands of frames. So I didn't go over it in this talk, but with localization imaging, we're turning on a small subset of molecules in order to find their center. And so each frame, we only turn on a small fraction. And so in order to get enough information, you need to take many, many, many more frames in order to get enough data to potentially make a time point. Versus with SIM imaging, we can go a lot faster. Or we can potentially go a lot faster, and we don't require as many images. So there are examples of live storm data or palm data in the literature, but they oftentimes are suffering from temporal resolution. So SIM is more suited for live cell because you can potentially get much faster frame rates.
Okay. Another question is, I am uncertain of using deconvolution. People in my lab don't seem to trust it as they think it is manipulating the image. What's your thoughts on this?
Yeah, there's a lot of some people have a lot of apprehension about using a mathematical process to get a final image out. What I recommend in that case to help convince people that it's not doing something funky to your image or producing something that's not real is to have appropriate controls. So appropriate control, for example, could be microtubules. We know those are the structure of microtubules are very well established. You can also embed your own beads into your own sample and reconstruct that. And if you see that the beads look as we would expect, then that can help you feel more confident with your data. And another method is to just compare to other methods. So if what you're seeing by deconvolution or potentially structured illumination also appears in other methods, that's a good indication that this is not manipulating the data or just some kind of artifact.
Okay, thank you very much. So the next question, it relates to something that I do believe you touched on, but I'm not sure if the viewer maybe missed that or maybe it needs further explanation. It says about oils, it seems like for each wavelength, one should use a different oil. Is that correct? What do we do if one sample has a few markers and I'd rather not move the slide to change the oil?
So yeah, I touched on this very briefly. What you can do is try to find an oil that is the best for the most wavelengths or results in the fewest artifacts for most of the wavelengths. And generally speaking, if you let's say you have green, red, and far red in your sample. If you match the oil for far red, the green and the red tend to be a little bit more forgiving.
Okay. And the next question is, can you quantify fluorescence intensity using SIM between different samples that may be at different levels from the cover slip? So for example, in a tissue.
That's a really great question. And I'm not sure. I don't want to say yes or no. I think there are some experiments that could be performed to help get at if this information. So in general, we do believe that our structured illumination algorithm is linear. But to help prove that, I think an experiment that would need to be performed would be imaging, let's say, a known sample at different fluorescence intensities, in proving that this is indeed linear or has a linear response. And then to follow up on the question in terms of different depths, I think that would need to be performed at different depths as well. So I guess the summary is I don't really know.
Okay, thank you very much. What is the thickest Z stack possible with SIM?
This is highly dependent on the sample. On the OMX, the working range of the objective is less than 30 microns, so there's no way we can go beyond that. You'll see several examples of 20 microns and very nicely prepared tissue section samples. And I would say it's generally more around 10 to 15 on pretty nice samples.
Okay, and another question, what's the fastest acquisition speed possible with SIM?
So I can't give you any specs on the other instruments or any custom built instruments, but on the OMX, with a CMOS camera, for 3D SIM, you can image at about 1 SIM frame per second. So that's 135 images in 1 second. For 2D SIM or turf SIM, we can go a lot faster than that. And it's always going to be dependent on your exposure time. So if you have a very long exposure time, you won't be able to reach those temporal resolutions. But I'm comfortable saying with a 1 millisecond exposure, for 3D SIM, you can do that single time point in 1 second.
Okay. So another question is the refractive index mismatch a larger issue for live cell SIM when the sample is in aqueous medium, and can this problem be minimized?
Yes, so you can accommodate or kind of correct for that or minimize that spherical aberration caused by mismatch by using different RI oils. So for example, on the OMX, we give a kit of different oils with a lot of different refractive indices, and so you'll need to adjust it accordingly to your mounting medium. So all those samples, or sorry, all those live images that I showed you were imaged with an oil immersion objective with the oils, the immersion oil optimized for the particular sample.
Okay, I think that's all we've got time for today. So it just takes me to thank you, Patrina, again for a really illuminating presentation and a wonderful discussion. And thanks also to our sponsors, GE Healthcare. And finally, thanks to you, the audience, for taking the time to attend and listen in. If you've enjoyed the seminar today and would like to view the video recording of the session, please visit the webinars page on bitesizebio.com. It should be available within the next 24 hours. There you can also see other webinars we've lined up for you in our Bite Size Bio Webinar Festival. So until next time, good luck in your research and goodbye from all of us at GE Healthcare and Bite Size Bio.